introduced the plant Angelica keiki, known across Japan as Ashitaba, or tomorrow's leaf. The rule of thumb for this coastal biennial is simple. Cut the stem today, and a new shoot will aggressively regenerate by tomorrow. During Japan's Edo period, political exiles banished to the remote island of Hachijojina faced severe malnutrition and compulsory labor, yet historical records note their extraordinary physical stamina. Their primary survival staple was this wild coastal plant, which they foraged to replace standard leafy greens. Today, Hachijojima is a demographic anomaly. Often cited by gerontologists as an island of longevity, it boasts a disproportionately high population of centenarians and healthy octogenarians. This combination of agricultural resilience and localized human longevity has fueled a booming functional medicine market. Ashitaba is heavily marketed globally as an anti-aging superfood, with aggressive claims that its compounds induce deep cellular cleansing, a process known as autophagy. The plant's historical utility as a survival food is well documented, but modern supplement marketing has generalized regional folklore to imply specific biomolecular mechanisms, creating the illusion of settled gerontological science. The wellness industry promises that consuming concentrated Ashitaba powders will directly extend your lifespan and actively delay the deterioration of your cells. A rigorous audit of the human clinical literature reveals a massive disconnect. There is zero human data testing Ashitaba for lifespan extension, incident age-related disease, or validated biological age measures. This visualization contrasts the marketing with the data. On the left, steep preclinical survival curves in yeast and flies. On the right, the strongest human evidence, the 2024 trial showing a moderate reduction in visceral fat over 12 weeks. Reducing visceral adiposity is a highly valuable clinical outcome for patients with metabolic syndrome, but shrinking belly fat involves biological mechanisms distinct from those required to slow the intrinsic hallmarks of human aging. The gap between anti-aging hype and metabolic reality reflects a chemical conflation. Practitioners and consumers are attributing the biological effects of two entirely different molecules to the same plant source. Prescribing generic Ashitaba is biologically imprecise. The plant yields a highly complex matrix of secondary metabolites, and the chemical payload changes drastically depending on how the plant is extracted. The primary commercial fraction driving the research is this thick, viscous yellow sap that aggressively exudes from the severed stems and roots. This sap is uniquely concentrated with a subclass of open-chain polyphenols called prenylated chalcones, specifically 4-hydroxydericin, or 4-HD, and xanthoangelol, or XAG. Look at their molecular architecture. These lipophilic prenyl side chains allow 4-HD and XAG to rapidly intercalate with lipid cell membranes. This feature drastically enhances their systemic bioavailability and cellular permeability compared to standard polar flavonoids found in other leafy vegetables. These specialized sap extracts are chemically distinct from whole leaf powders or green juice preparations, which mostly deliver basic dietary fiber, vitamins, and an entirely separate class of botanical compounds. One of those distinct leaf compounds is a highly touted flavonoid called 4,4-dimethoxychalcone, or DMC, which is found primarily in the aerial leaves and generic unrefined sap mixtures. DMC is structurally distinct from the 4-HD and XAG found in the pure yellow sap. Standard commercial sap supplements are standardized specifically for the sap chalcones and do not guarantee their DMC content at all. Failing to differentiate the leaf-derived DMC fraction from the sap-derived 4-HD fraction is the root of the clinical confusion. Consumers are swallowing sap extracts while expecting the biological outcomes of the leaf. To understand the origin of the anti-aging claims, we have to isolate the DMC molecule. This compound was the exclusive focus of a landmark 2019 paper in Nature Communications that single-handedly ignited the Ashitaba longevity hype. Researchers discovered that isolated DMC functions as a potent caloric restriction mimetic. It chemically tricks the body into a state of nutrient scarcity, triggering massive systemic macroautophagy to degrade and recycle aggregated cellular debris. This signaling diagram maps the mechanism. Historically, recognized gerotherapeutics like rapamycin induce autophagy by directly inhibiting the mTORC1 signaling pathway. DMC bypasses the mTORC1 cascade entirely, operating through a distinct parallel route. Instead, DMC targets and activates specific GATA transcription factors. This triggers a conserved metabolic reprogramming, altering cellular amino acid composition to force the cell into a defensive, self-renewing survival state. The evolutionary conservation of this papwini yielded profound results. Administering isolated DMC resulted in statistically massive lifespan extensions across entirely different biological kingdoms, including yeast, nematode worms, and fruit flies. In mammalian in vivo models, isolated DMC provided intense organ-specific cytoprotection, explicitly shielding mouse cardiac tissue against severe necrotic damage during prolonged ischemia. But here is the crucial limitation. That powerful geroscience signal belongs exclusively to isolated, purified DMC, utilized in non-human models. It does not apply to the generic Ashitaba powders or sap capsules available to consumers. DMC is a highly intriguing mTOR-independent autophagic agent, but it remains an experimental preclinical compound, not a validated human gerotherapeutic. We must pivot back to the sap-derived prenylated chalcones, 4-HD and XAG. These are the molecules driving the standardized products, and they are the actual drivers of the human clinical data. This model of an adipocyte illustrates their primary intracellular mechanism, the direct phosphorylation and activation of AMP-activated protein kinase, the body's master energy sensor. When cellular energy drops, AMPK forcibly shifts the cell's machinery away from anabolic fat storage. 4-HD and XAG manually trigger this response, suppressing master lipogenic transcription factors like SREBP1 and PPAR-gamma. This mechanism acts as a potent anti-adipogenic agent. It prevents pre-fat cells from maturing into lipid-storing adipocytes, while simultaneously inducing the translocation of GLUT4 transporters to pull glucose out of the bloodstream. This cellular mechanism translates clearly into human evidence. A 2024 randomized, double-blind, placebo-controlled trial tested encapsulated sap chalcones on mildly overweight adults. This chart reconstructs the primary clinical finding. Utilizing just 16 milligrams per day of total chalcones, the active group achieved a statistically significant reduction in computed tomography-measured visceral fat area over 12 weeks. Related pilot studies tracking metabolic syndrome parameters have also noted favorable modulations in fasting ghrelin, the body's primary hunger hormone, providing a neuroendocrine mechanism for appetite suppression. Standardized Ashitaba sap provides a targeted adjunctive metabolic modulation that improves lipid handling and visceral adiposity. Because these sap extracts exert powerful biochemical influence, we must evaluate the toxicological safety profile and highlight the explicit clinical contraindications. The baseline safety is highly reassuring. 90-day GLP rat studies demonstrate a massive no observed adverse effect level of 300 milligrams per kilogram per day, indicating broad systemic safety even at high concentrations. We must also correct a widespread nutritional myth, heavily exploited by alternative medicine, the assertion that Ashitaba leaf can reverse clinical vitamin B12 deficiency in vegan diets. Advanced mass spectrometry reveals that the plant primarily contains biologically inactive pseudovitamin B12. This analog actively blocks true cobalamin absorption by competitively binding to transcobalamin transport proteins. But the most severe clinical contraindication for Ashitaba sap extracts is directly related to its potent influence on the cardiovascular and hemostatic systems. As shown here, both 4-HD and XAG forcefully inhibit the synthesis of thromboxane B2, making them powerful systemic antiplatelet agents. Concurrently, plant furanoplumerins and chalcones can act as competitive inhibitors of the CYP2C9 enzyme in the liver. That liver enzyme is critical for metabolizing oral anticoagulants like warfarin. When the enzyme is inhibited, the drug's serum half-life violently spikes, amplifying the anticoagulant effect. Combining reduced warfarin metabolism with direct platelet inhibition creates a severe synergistic bleeding risk. Prescribing high-dose Ashitaba extracts to metabolic patients on blood thinners risks uncontrollable fluctuations in INR and spontaneous hemorrhagic events. To evaluate its true utility, we have to objectively position Ashitaba within the broader landscape of modern geroscience. Ashitaba possesses zero human aging data. It cannot compete with foundational interventions like vigorous exercise and caloric restriction, which boast massive, irrefutable human cardiometabolic and mortality outcomes. This chart ranks the evidence hierarchy. Pharmaceutical gerotherapeutics like rapamycin and metformin have vastly deeper mechanistic roots and broader epidemiological datasets. In the context of longevity, Ashitaba sits near the bottom. The island of longevity phenomenon in Hachijojima is a real demographic event, but isolated regional longevity is heavily multifactorial, relying on genetics, constant low-grade physical activity, and community structure. It cannot be chemically reduced to a single botanical extract. If the goal is prioritizing lifespan and healthspan, Ashitaba is drastically outpaced by strict cardiometabolic care and top-tier geroscience candidates. The clinical utility of Ashitaba requires a strict deductive approach. Its pharmacology is split between two distinct pathways, and treating it as a single generic entity is scientifically invalid. The leaf-derived DMC molecule remains a highly promising mTOR-independent autophagic agent, but it is strictly confined to the preclinical realm. The sap-derived 4-HD and XAG chalcones possess moderate RCT-backed clinical utility for targeted visceral fat reduction and insulin sensitization. Clinical professionals must demand precise chemical standardization in commercial formulations. You have to ensure that the specific molecule you are prescribing actually matches the intended outcome for your patient. Ashitaba is a fascinating, bioactive metabolic botanical with genuine utility in lipid management, but it has not yet crossed the threshold from plausible nutraceutical to proven gerotherapeutic.